Thank you, Joe. Thank you very much, Joe and Professor Acambo, for this wonderful invitation at this terrific inaugural conference. I was just going to make the motion, since speech doesn't do it like music does. Maybe we'll forego this talk and just have Judith come back and sing a little more. What do you think? Can we have a show of hands for Judith? All right. How many of you were here yesterday at the ABT conference? Oh, terrific. So we have a little bit of context. It was a terrific conference with Alex and Don Campbell. What I put up in the slide here is a photograph from Time Life, which was one of their 2001 Time Life photographs of the year. And it was taken within a week or so of the United States Army driving the Taliban out of Afghanistan. And to the right here, you see a mob going to the cinema in Kabul for the first time. This is the first time in 13 years that women were allowed to dance or sing. So this is a young woman at her wedding a few days in Kabul after the Taliban were driven out. And I show these slides in particular to remind us of the universality of music, the universal competence of humans to apprehend emotion and meaning in music. There is no culture that's ever been known, either present or past, to be without music. And when we start to talk about the biology of music, we're particularly interested in establishing the universality of different aspects of music perception and cognition among listeners, and also innateness. Those two are key foundational elements of the biology of music, the universal competence in apprehending emotion and meaning, and the innateness of our ability to acquire effortlessly and without conscious effort the syntax of music, the phonology of music, many of the things that we say as we apply it to language, or that Noam Chomsky would apply to language, we can apply to music, although in a different sense. Now the next slide, I'm going to do a little tachistoscopic presentation. Hopefully we'll get to split brain experiment, which involves using a T-scope. So I'm going to flash a picture to you and then ask you if you recognize it, okay? We ready? Okay, anybody recognize that picture that flashed by there, Vera? Vera, what, you recognize that at all? Yeah, album cover of Sgt. Pepper's, Dr. Campbell.
There was a catalog done with a parody on that had Larry Dossey, a neurologist and musician.
Yeah.
And looked like that to me.
Okay, so this is the back cover of the Molecular Biology of the Cell, edition 3. And they do a series, like each edition is a different Beatles album cover. There are a lot of Beatle fanatics in the fan. I happen to be one of them. I don't think I'd be doing this if it weren't for the Beatles. And I learned at dinner last night just how many of us have been influenced by them, even generations after mine. But this is the president of the National Academy of Sciences in Washington, who's the lead editor. And I just bring this up to you, well, for two reasons. One is music permeates science and medicine. There are so many scientists and so many physicians who have a music background and a love of music. So in many ways, those of you who are thinking about making inroads into the fields of medicine and science with music, you have a little bit of a captive audience. There are a lot of people in the field who really have a love for music. And most of us, if not all of us, who've contributed to the field do have a music background, although the number of rockers and jazz musicians are precious few, mostly classicists, as you might expect. And the second reason I show this slide is to thank the members of the board of the Institute for Music and Brain Science. And yes, Don, it's easy to put people's heads on these things. Ann Young, one of the founding members and the chair at the Department of Neurology at Massachusetts General Hospital. I think my laser pointer is going. If you have a backup laser pointer, I could use one. Oh, here we go. That's Ann here. David Hubel, who won the Nobel Prize, I actually didn't have to superimpose his face. It was there. George Martin, the Beatles producer, a member of our founding board, Jamie Singleton, Caroline Beanstalk, who is the COO of the world's largest independent music publishing company. And without the support of these board members, I wouldn't be here today, and I also want to acknowledge the Grammy Foundation for supporting some of the research that we've done at the Institute. One of the interesting, thank you, Joe. When we talk to neuroscientists about, why study music? Why look at the neuroscience of music? Well, in many ways, it grew out of the subdiscipline of neuroscience called cognitive neuroscience. And if we look at the brain through the perspective of music, we can address many of the things about brain function that other areas of cognitive neuroscience can. Auditory perception and cognition in particular, motor skill acquisition and performance, multimodal integration, learning and memory, communication and semiotics, or the study of meaning, emotion. But we go a little bit beyond, I think, in music. We offer a little bit more to the field of neuroscience because we can start to address questions about aesthetics, about talent and creativity. Those are really things that have been outside the reach of neuroscience, and it's really only by studying the arts from a neuroscience perspective that we can begin to talk about a neuroscience of aesthetics, a neuroscience of creativity. So it's a very rich medium for applying the scientific method to learn about all of these higher brain functions, and in addition, the brain functions that apply to the arts. And this has helped to fuel the interest in the field over the past 10 to 20 years. I got into it because I was interested in auditory perception via music perception. And if you're interested in hearing, it's not just about perception, it's about auditory cognition, how context influences speech perception, how context influences music perception. And in the nonverbal domain, music is, like environmental sound perception and recognition, is really what we have to study nonverbal auditory processing. In a sense, tonal information processing, which is how I originally got into the field. And of course, there are implications for the diagnosis and treatment of brain and ear diseases, which we heard a lot about yesterday, and we'll hear more about today. Yesterday in the ABT conference and today in this Quantitative Research in Music and Medicine conference. Now, it's not as simple as the old phrenologists in the 19th century had led us to believe, or at least people at that time to believe. When we talk about the functional organization of the human brain, a lot of it gets into the world of localization. All right, and you see a lot of these colored images now in the press with fMRI or PET scans. If you look at stroke images, there's a motif within the field about where is a particular brain function. Of course, the phrenologists, they said tune was here in the right or left frontotemporal region. Oddly, they weren't too far off, but this sort of application doesn't apply. And because this is a quantitative research conference, I want to go through some of the approaches over the past couple of hundred years that have been used to try and come to some conclusions about brain organization in relation to function. So first of all, we have anecdote. Oh, I had this patient, and it's sort of Oliver Sacks' Tales of Music in the Brain. Oh, I had this patient, I saw this, so it must be that, right? In today's century, that's not acceptable, okay? It's useful because you have to have a rational approach to generate working hypotheses that you can test experimentally. It's not that there's no role for rationalism or anecdote, but in today, what's required of us today, if we're going to make headway in this field, is to get away from these old-fashioned methods. Another one is introspection. So George Wundt, even the great Hermann Helmholtz, in his book on the sensation of tone as a physiological basis for the theory of music, he used introspection. You know, I played this interval to myself, it sounded dissonant, roughness is the basis of dissonance. You know, that doesn't fly anymore. You have to do a study with unbiased listeners to get at that. What the current scientific method, the philosophy of science, involves are two basic principles. One is called verificationism, and the other is called falsificationism. Falsification theory being proposed and popularized by Karl Popper, an Austrian-born English philosopher. And the idea of falsification theory, you may know it in terms of the null hypothesis, is before you do the experiment, this is the biggest sin of all the functional imaging, or many of the functional imaging experiments. Before you do the experiment, based on rational grounds, intuition, introspection, and so forth, you clearly state a hypothesis. For example, we're going to look at today, a hypothesis would be the auditory cortex is essential for normal pitch and harmony perception. And then one has to go and do an experiment, make planned observations in a controlled environment that attempts to disprove your hypothesis. And if you fail to disprove the hypothesis that supports the hypothesis and allows me to say the auditory cortex plays an essential role in pitch and harmony perception, that's the approach. And as many of you know, this has not been the approach when we talk about music therapy and music-related therapy. And this is exactly what Professors Akambo and Rosowski are trying to get at, that to develop a society and a field and a track whereby this sort of philosophy of science is applied to the therapeutic uses of music and listening. So testable hypotheses are good. Of course, you may know from the pharmaceutical literature, the drug development approach is something called the randomized controlled clinical trial. And I'll show you a randomized controlled clinical trial that my group did with premature infants and how we went about the methods of doing that. The basic idea is that you have to have a test group where you're testing your testable hypothesis, and then a control group, for example, there's one way to do it, a control group that's similar to the test group in every way, in every possible way, and you have a number of predefined measurements that you're going to make after testing the hypothesis, and then compare the results from the control group to the test group using statistical methods to show a certain amount of probability that your hypothesis has been proven. All right, so it's much more difficult and doesn't exclude the one-on-one psychotherapy type of interaction. That's important in its own right. But if we're going to start getting third-party payers to support the kind of work that we want to do, if we're going to publish our papers in peer-reviewed journals that are read by physicians who are often in positions of authority to enact the kinds of treatments that we're trying to promote in this conference, we have to play by these rules. We're not going to make any inroads unless we test hypotheses, we do randomized controlled clinical trials, and we publish them in peer-reviewed professional journals. Now, what are peer-reviewed professional journals? For those of you in music, it's sort of the equivalent of the audition. Anonymously, 2 or more experts read your paper and tear it apart. And you have to survive that criticism, revise your paper. If you don't do it adequately, you do not get published. You may go to another journal and try them, and there is a pecking order of professional journals. So, you know, getting a music-related study into the New England Journal of Medicine would be sort of the holy grail. Right? That's not going to happen right away. And there are ways of dealing with this. We mentioned yesterday that there's now a new journal that Sage Publishing put out called Music and Medicine. So they're looking for the kinds of studies that we're talking about that are done well, And they, in their own right, can publish those papers. So we have to make inroads using these techniques. And it's important in a multidisciplinary meeting like this, where we all come from different backgrounds, that we at least have a little bit of a common ground about the philosophy of science and how we're going to move a field from the arts into science and medicine. Now, I have to go through, again, for common ground purposes, a little bit of brain anatomy. And what I will not do is enumerate the 40-plus areas of Brodmann and tell you where they are in the cerebral hemispheres. I never found this useful as a student. But there are other reasons that this doesn't apply. One is that it's a purely anatomical method. So what Brodmann did was take a dead brain, slice it up into microscopic sections, look at it under the microscope. Some cells look like triangles, some cells look like circles. There are 6 layers, and you say, well, are the big circles and big triangles in layer 4 or 3? And how many different areas can I identify based on the distribution of triangles or pyramidal cells and circles or granular cells in the 6 layers of the cerebral cortex? That's the game. That's the science. Okay, and it's a very important piece of microanatomy, and it carries this great term, cytoarchitecture. It sounds really important. But it's actually not useful when we talk about function. For example, when Economo and Koskinas broke up the brain and looked at it under a microscope, they came up with 107 areas. So this is the problem of the eye of the beholder. We want to get away from the problems, even within science, of sitting at the master's feet and reproducing what the master does and doing it in the eye of the beholder. And furthermore, what we're really interested in is how anatomy relates to behavior. It's going to be hard to get that out of a dead brain, right? So I've never really found, although in certain ways it's useful in certain aspects of neuroscience, for our purposes today, I don't really think we should go through the 4 dozen Brodmann's areas. What we can do is talk about the cerebral cortex as an amazing computational machine. The cerebral cortex is, if you imagine aluminum foil, right? A piece of aluminum foil that's as big as, let's say, a small Domino's pizza. All right. Now, although it's thin, that sheet of aluminum foil does have a thickness. And you can wrinkle that aluminum foil up, right? And by wrinkling it up, you can fit more aluminum foil in a Tupperware than if you didn't fold it up, right? Well, that's what evolution has done with the human cerebral cortex. You know, we are the kings and queens in the animal kingdom of the human cerebral cortex. So it's a 3-millimeter thick sheet, okay, the size of about a small Domino's pizza. And in that area and thickness, there are approximately 20 billion neurons, or the information processing units of the brain. And each of those neurons, on average, makes about 1,000 connections. So there are 20 trillion connections. We used to joke in the lab about Carl Sagan and the cosmos. That there are billions of, right? It's kind of the same thing for the brain. There are 20 trillion connections in the brain, right? And you're making them all the time and losing them all the time. The whole use it or lose it principle in neurobiology relates in many ways to the cerebral cortex and those connections that we're making all the time. We really don't make many neurons past about mid-gestation to late gestation as fetuses. Making neurons is not what we do, but we do make connections and do prune those connections. One of the problems with autism has to do with pruning at age at 2 years old, but still have this enormous. So if you can imagine the computational power, there's no computer that can do with sound what your auditory system can do. All right, or with vision that your auditory system can do. And the seat, if you will, Aristotle had it wrong. He thought it was the heart, and he was very disrespectful because Hippocrates was 200 years older than Aristotle, and he'd already done lesion effect experiments. The seat of intellectual and aesthetic life, for the most part, is this cerebral cortex. Fortunately, so on the right you have a normal, on the left you have an Alzheimer's disease brain, and you can see the atrophy in the Alzheimer's disease brain. And the Alzheimer's is a disease of the cerebral cortex. Now, fortunately, there are only so many lobes. There's a frontal lobe, there's a temporal lobe, there's a parietal lobe, there's an occipital lobe, and then the structures underneath, which I'll call the subcortex. So rather than remembering all these different Brodmann's areas or gyri, let's try to stick with the basic lobes of the brain. And the way they're defined as lobes are not by these bumps. They're called gyri. How many of you have ever had like a Greek hieros? It's the same root of the word. So you know how the gyros is like a rolled up sandwich? So these folds on the brain are called gyri. But it's these crevices between them, or sulci. So there's a big one here that separates the frontal and the parietal lobe. There's a big one here that separates the temporal from the frontal and parietal. And there's an imaginary one here that separates the occipital from the parietal. All right, so that's how when we look at the brain, if we have a model of the brain and we're to name these four lobes, if you can remember those four, it'll facilitate a little bit of what I'm going to talk about with functional brain organization. Now, I've tried to simplify this further from a functional perspective. Remember, those are still anatomical terms, right? They're about areas on the brain that you can give a name to for whatever that's worth. But really what we're interested in is how those areas function. And those types of brain areas, those functional zones, if you will, can be broken up into three basic types. There's the modality-specific type, which here is in red and blue. I don't know if we can get the lights down a little bit for the slide so that we can see at least the slide a little bit better. So these modality-specific neurons within the cerebral cortex, they're devoted to processing information in only one sensory modality or for movement. So there's a visual, there's an auditory, there's a touch, there's a movement area. So if we just keep that in mind, there are these modality-specific areas. But of course, we don't perceive the world, at least non-autistics, don't perceive the world as Humpty Dumpty, this incoherent, separate. I mean, think about your body, right? Your eyes are over here, your ears are over here, your skin is over there. So how do you put all of that together into a coherent whole of your body for sensory experience and movement? How does that happen? Well, one of the two major theories is that there's a convergence of these neurons. They send their connections to other neurons that are multimodal. So to excite those neurons, they'll only fire if you have a visual stimulus and an auditory stimulus co-registered. An example of this for all of you, very, very straightforward actually, is when you read. Don't you hear yourself read oftentimes? So you're putting together the sound of the word with the sight of the word. That's multimodal processing, right? And in fact, phonological processing deficits are the main problem for reading. Their auditory processing deficits are the main cause of dyslexia in children. It's actually not just reading the words, it's putting those symbols together with the sound. And of course, we don't have to be taught how to talk or how to do language. We do have to be taught how to read. Reading has only been around for, writing has only been around for about 4,000 years, but we've been talking forever. So that's another example. How many of you play a musical instrument? Okay, so that's a really clear example where you have the instrument. Think about, like imagine Judith up here, right? There's the movement of the hands and vocal cords. There's knowing spatially where your fingers are on the fretboard, right? Moving the fingers on the fretboard, hearing what you're playing, modifying what you're doing based on what you're hearing. And for those of you who read, you're also doing visual processing. And one of the reasons we talk about music is a great way to exercise the brain is because you're involving all of these modality-specific systems, including the vestibular system and the multimodal systems. So you're getting a lot of the brain activated when you're actually doing music, right? Even when you're dancing. Now the third area here that I called supramodal is really a relatively, at least a part of it is unique to humans. That is the processing of information above and beyond stimuli from the external world. So a lot of our internal lives, right? Who we are, really, we can all see, we can all walk, we can all hear, but we identify our individuality in relation to these supramodal areas, our memories and how we plan for the future, how we interact with society. In other words, supra is Greek for above or beyond. So this is the processing of the brain that goes above and beyond stimulus modality, above and beyond movement. So I always say the 7 deadly sins and the 7 virtues, right? They're in the domain of the supramodal cortical areas. And they're not, and we've developed them evolutionarily way beyond any other animal, including chimps, who we've separated from only about 7 million years ago. So it's hard to get philosophers to agree, but I think that most, if not all of them, would agree that there are 2 distinct realms of everyday experience: the inner self and the external world. And the photographs I have here are John Locke, the great 17th century British philosopher, on the left, and Immanuel Kant, the great 18th century German philosopher, on the right. So what nervous systems have done is to evolve specialized mechanisms for interfacing the inner self and the external world. So we have stimuli that impinge upon us from the external world, your eyes, your ears, your skin, and so forth. And those are all over different parts of the body. They get processed in the peripheral nervous system and spinal cord. Like the deep tendon reflex just goes right from the nerve to the spinal cord, back to the nerve, and back out to the muscles, bones, joints, and so forth, so that we can exert the force of our actions into the external world. So this is what's happening at the interface with the external world. So if we go higher in the processing of information in the nervous system, we come to that area I called the subcortex, or S. And the subcortex is reciprocal in that it feeds back down to the peripheral nervous system and spinal cord. And then the next level of processing within the system are these modality-specific cortical areas that I was mentioning: auditory, visual, tactile, motor. Those modality-specific areas project to the multimodal cortical areas that put together the different modalities and help solve the Humpty Dumpty problem. Okay? Now, I put a line here between the multimodal cortex and the inner self because, again, there's nothing particularly characteristic about each of us with respect to these other types of processing. And the multimodal cortex then projects higher. There's a concept here of lower and higher. Binary opposition, so important to structuralism and science and understanding how everything from different societies to the brain works. So there's a lower, higher dichotomy here in binary opposition. The highest level of processing is the supramodal cortex. And this is what John Locke, this processing, there's a jargon within psychology and cognitive neuroscience. This is called bottom-up processing. All right, you're taking information from the external world, and in serial stages, you are processing that information within the brain. And what Locke said in his essay on human understanding in 1691 was that at birth, the human mind is just a blank slate, or tabula rasa, and can only acquire basic knowledge of the world through the senses. After this initial process, experiences can be categorized and stored in the memory. Now, this whole bottom-up approach isn't as simple as I've laid it out to be or as it was thought to be, say, in the middle part of the 20th century. The supramodal cortex doesn't only get inputs from multimodal cortex. It also gets inputs from modality-specific cortex and even from the subcortex. So this straight line from the external world to the supramodal cortex, that bottom-up processing is called serial processing or hierarchical processing, because we're positing that the supramodal area is higher in the processing chain than the lower areas, so that's serial or hierarchical processing using a bottom-up approach. But there is parallel processing, because the inputs to these higher areas aren't just serial, they're also parallel from other levels of the nervous system. This has been a major theme in basic neurobiology research, is to try to differentiate what the contributions of serial processing or hierarchical processing versus parallel processing is, whether it's music, whether it's vision and color, whether it's consciousness. There's an interest in understanding why we have both serial and parallel processes. But you can probably tell from the way the arrows are placed in the slide that it's not all bottom-up. And Kant is in many ways the founder of cognitive science. He was an empiricist like Locke. But what Kant said was that the mind organizes and systematizes what we experience with its own programmed intuitions and categories, and that we need those categories and intuitions to make sense of all of the data that is constantly flooding our senses from the external world. So if we weren't chunking the information, if we weren't parsing it, again, dichotomies, binary oppositions, an important way that we use to understand the world, right? Breathe in, breathe out, left, right, good, bad, north, south, right? Categorizing information is essential to actually understanding processing in the external world by the inner self. So what Kant is emphasizing, and what the cognitive psychologists over the past decade or two have really talked about, is top-down processing, right? This is the notion that what you know about the world influences how you perceive it. So there's no human perceptual experience that is without bias, because your knowledge of the world, whether it's implicit or explicit, whether you can articulate it or not, is going to influence how you perceive stimuli coming at you from the external world. I remember when Leonard Bernstein gave his Norton lectures at Harvard in 1973, and he was talking about a musicolinguistics. He said, well, we can all understand maybe emotion and meaning in Western music because we have knowledge of the idioms and so forth that we use in Western music. But can we really make sense and extract emotion and meaning from an Indian raga, or music that we're hearing for the first time? We really don't have knowledge about that genre that will allow us to shape our perceptions of the stimuli that are impinging upon us. So, this is a way of introducing and getting everybody on a common ground that we've covered some really basics about the brain. We can name places in the brain, like the frontal lobe and the temporal lobe. That's anatomy. Okay. We can talk about what the brain does or different parts of the brain do. That's function. That's the mind. Okay. What we don't talk, and we won't talk too much about today, is the chemistry of the brain. That's the third major area of inquiry, is looking at brain chemistry and how it relates to behavior. Okay, and of course the parallel systems work top down in the way that they do bottom up. All right, so what I want to do now, because it's so relevant to the health and medicine aspects of this conference and the previous day, is to focus more, at least for the time being, on the supramodal areas. And what I've done is to break up the supramodal areas into a supramodal past and a supramodal future. So I had this idea. Of course, one of the dangers of reading is that you realize you never have an original idea. So I went back and I found some work from Parmenides in the 6th century BCE, and interestingly, Augustine, and the quote from Confessions in the 5th century is, "Neither things to come nor past are. If times past and to come be, they are not as future or past, but present." So the concept here is that the only thing that exists is the present. You know, Augustine did some self-flagellation and apologies to God in Confessions after he said this. "Oh, I'm sorry, God." You know, but the notion here is that the only thing that exists is the present, and it's a continuous flow. The past and the future are figments of our imagination. They exist only within the inner self. They're not there in the external world at all. So these areas in the temporal lobe, this medial portion of the temporal lobe, I don't know if you can make out these folders, right? Those neurons bring the past into the present. So memories, episodic memory, for example, feelings, that's bringing your personal history or our evolutionary history to some extent into the present. So those are the supramodal areas that deal with the past. What humans are, to a fault, really good at, and way beyond what any... I got very close to some old world monkeys when I was doing my thesis. They are so stimulus driven. It's like they really don't have much of an inner life. At least it doesn't appear to be. They're very stimulus driven. What humans excel at is bringing the future into the present. Planning, strategy. And I think about coming to the conference. You know, you have to get your tickets, you have to clear your schedule, you have to pack your bags, what are you going to take, why are you going, how much is it going to cost, all that. But for a conference that's going to happen like months, David, it's like years, you know, and Joe. So you're bringing the future into the present, and those areas are mediated by the front of the frontal lobe. You can see these hourglasses here, which are bringing the future into the present. Now, if we did comparative anatomy on an evolutionary tree, monkeys have a very small, it looks like they've had a frontal lobotomy compared to us. Apes, it's a little bit bigger, but it's still nothing like the human frontal lobe. And you may hear this term sometimes in the literature. People call it the prefrontal cortex. So the prefrontal cortex would be over here. There's nothing in front of the frontal lobe, okay? It's a term that what people mean is really, there's motor, there's premotor, and there's premotor. But nobody wants to say premotor, so they say prefrontal. Premotor, so they say prefrontal. It's just it's one of those great, awful terms in medicine and science. It's really the front of, or anterior portion of, in Greek, the frontal lobe. And those two areas are very important when you think about using therapies on patients. A lot of it is not just the present, but aimed towards working with their past or moving towards the future and how you're going to make them better. So it's very important to address these supramodal areas. Okay, so how many of you have ever written a song or composed a piece of music? A lot of you. It's just totally different from a neuroscience conference, which is where I'm usually talking. It's like nobody raises their hands, right? Including the musicians in neuroscience, because mostly they've been drilled on Suzuki or classical piano to get into med school or something. So the holy grail for any sort of composition, it's not about all of these higher level functions, right? It's about evoking emotions and their visceral concomitants. In other words, increasing someone's heart rate, getting them sexually aroused, whatever it may be. This is the holy grail. So when I wrote a review for Science about how might it be that we derive emotion and meaning from music from a brain perspective, well, of course, for music, we want to understand auditory analysis and representation. So the yellow coating here you see on the temporal lobe, these yellow areas, those are the modality-specific auditory areas. And much of what the research has shown is that for the perception, the analysis, the representation of pitch, harmony, octave equivalence, equivalence and transposition, that's the work of the auditory cortex, the modality-specific auditory cortex. So how do we go from those auditory representations to emotion and meaning? How many of you have come across Leonard Meyer's Emotion and Meaning in Music? It's a classic book from 1956, now out of print, but something I would highly recommend to you. Leonard didn't come up with these terms, but he did formalize thinking and popularize the thinking about emotion and meaning in music, and he posits two main routes to deriving emotion and meaning from these auditory representations. One is the absolutist route, or the intra-musical route, and the other is the referentialist route, whereby you make semantic associations in the true sense of Saussure's signs, that you're signifying through music some other event. All right, so let's take a look at these. So the absolutist route, one of the dominant themes is expectancy, generation, violation, and satisfaction. In music, we talk about repetition and return. We talk about appoggiatura and cadences. You know, they're really some of you could make these really corny examples about expectancy generation. So, I mean, you all know, just to demonstrate what a cadence is, you know, at the end of most classical pieces, you hear. But I have to end here. Right? If I ended here, it's not quite as resolved. I'm not satisfying your expectancies as much as if I end, right? So that's a trivial example. Appoggiatura is probably one of the most interesting examples where you really violate, even in terms of key, what you hear and then quickly resolve it, okay? And in many ways, what you're doing there, you can see this is colored purple because the anterior, well, I guess we don't see the colors well on this projector, but this is supposed to be purple, this is purple. So you're really bringing the future into the present. Where is the melody going? Where's the harmonic progression going? Where is the rhythm going? Right? It's a continuous flow of bringing the present into the future. We had a great conversation with Dr. Belli last night about drumming and pulse and rhythm. It's sort of like the rhythm in many ways kind of carries you into the future. And then these melodic and harmonic, at least in Western music, and these harmonic and melodic expectancies are either satisfied or violated. Again, I'm using binary oppositions where probably I should be more deconstructionalist, post-structuralist, and say there are a lot of undecidables in music when it comes to violation and satisfaction. But for the time being, let's think of it from a structuralist standpoint, okay? It's this really abstract way of getting at the subcortex which is where we generate a lot of hormones and neurochemicals, where if you stimulate these areas in a rat and give the rat the bar will press the bar and press the bar and press the bar and die rather than eat. Right, the classic Olds-Milner experiment. So what the supramodal areas in that top-down projection are doing are going back down to the subcortex with some sign saying there's a positive or a negative emotion, and then generating excitement, changes in heart rate, and so forth, top-down. All right, what about the referentialist route? Well, the referentialist route has to do with associations with people and past events. So things that are very much related to cultural norms. The music, I mean, you would never play music from a holiday at a funeral, right? There's wedding songs, you know, oh, that was the song that was playing the first time I kissed a girl. Those sorts of associations where there's a reference not necessarily intramusical but extramusical to something that happened outside of the realm of music. And by activating those associations and representations, there are projections from those temporal areas that are part of the supramodal past system to the subcortex to again evoke changes in heart rate and so forth. Now, I think that this is actually an incomplete picture of emotion and meaning in music, and I don't know that we can blame Leonard as much because he was very pre-MTV, right? So, but he wasn't pre-theater, and he wasn't pre-dance. So the fact of the matter is that music is not only about hearing. And there are cultures, I was asking Professor Akambo about this, and Kenya is one of them, where there aren't separate words in the lexicon for music and dance. So when you hear a piece of music or you dance to a piece of music, your body is moving, right? What happens when you start exercising your body? Your heart rate goes up, your blood pressure goes up, you start sweating, and you feel those things. So there's a feedback loop that comes from your body back to your brain saying, I'm excited. So kinetics and kinesthetics, which really belong to the modality-specific areas having to do with movement and touch, those areas are involved as well, and they contribute to emotion and its visceral concomitants. Visceral concomitants, I mean, you know, heart rate, goosebumps, chills down the spine, those sorts of things. And similarly, visual perception, right? Stage design, scene lighting, dance, the way that you look. You know, Joe and I and a bunch of us here, we may love the Beatles' music, but when they made it, the look was important, right? It changed the way men wore their hair, and they wore high heels, albeit in suits and ties. This is, you know, now on MTV, it's like, you know, you don't wear clothes and you can sell records. So you have to look good, right? Looking good is part of selling the song. That's visual. So visual perception cannot be divorced from music processing in the holistic sense. So all of these different modalities are contributing to the generation of emotion and activation of these visceral concomitants. It's not purely the absolutist and referentialist route. I would think of them more as sort of the multimodal means of activating emotion and visceral concomitants. A tough nut to crack has been personality and preference. We tend to think, because of work done with lesion effects, I have a case, I don't think we're going to have time to get to it, of this area at the bottom of the frontal lobe here, which if you have damage there, your personality changes. You know, there's a famous case of Phineas Gage, who had a tamping iron go through his mouth and out. He was working on the railroad with dynamite, and it went, and it destroyed this part of the frontal lobe. And he went from being the foreman, who was really a terrific husband and worker and reliable, to basically joining the circus and leaving his wife. And so we think this has something to do with this, but we really have, I think, in neuroscience really don't have a good understanding about, there's basically in neuroscience it applies as in, in general, there's no accounting for taste. Right? It's probably the ventromedial areas. All right, so I just got the 5-minute sign, so I'm going to skip a lot of slides because I do want to get to some of the more health-related or medicine-related part of my talk. Okay. So, how do you get blood from a tiny baby? So, currently, in the United States and Europe, 12% of babies are born prematurely. So 1 out of 8 babies. And when you're born prematurely, you always have a blood test. I mean, even if you're not sick, which is what the study that we did in premature infants, they weren't critically ill or anything. But how do you get blood from a baby? Does anybody know? We didn't talk about in the car. Okay. The veins are too small. Yeah. Yeah. You grab the foot, you take a needle, you stab it in the heel and squeeze. It's like when I was a medical student and doing my peds rotation, it was like, what? Or you got to be kidding. And what do you think the baby does? Wah! So when Nightline covered our study, they put this baby here. This is a baby from the study, okay? It's not, Nightline didn't want to show this as, the picture to yoke up with doing this research. So there's a really big problem in neonatology of how to deal with pain and stress in premature infants. And don't forget the babies who are in the neonatal ICUs, they'll get a heel stick, 1 to 3 heel sticks a day. And you can't give them painkillers. You can't adequately numb the spot where you stab them. You have to squeeze for a while. I was interested in that from a medical perspective, but also from the innateness standpoint. A lot of what I've said about auditory perception and music and emotion and meaning has been demonstrated experimentally that you do not have to be a musician, of course, to apprehend emotion and meaning in music. It's universal. Every culture has it. So what about the innateness argument? Well, there was a study done in Boston a few years ago by Marcel Zentner and Jerry Kagan. Jerry's one who's like fans. He's like this hero of child visual perception, infant visual. The infants like round faces. That's Jerry Kagan. So Jerry had a postdoc come from Germany who said, well, what about music? And Marcel had a great idea. He had 2 types of stimuli. The way these experiments are done are, let's say you put a loudspeaker behind a bullseye. And the baby looks at the bullseye, and then you turn the music on. And you measure how long the baby looks at the bullseye. And also, if the elbows flex or the hips flex. So these are 4-month-olds sitting on their mother's lap. And Marcel had some great stimuli. He did basically lullaby-like songs. So one of the stimuli would be something like, okay, the other stimulus, and looking at the response of the babies, was, let's see if I can even do it, because my cognitive representation of this is so weak. You never play this. So one is with all thirds, major or minor, and the other is with all minor seconds, which are traditionally dissonant intervals. And what they showed that even by 4 months of age, the babies looked longer at the bullseye when it was the consonant version in thirds, but when it was the minor second version, all hell broke loose. The elbows flexed, the hips flexed, they cried, they turned away from the thing. So by 4 months of age, they demonstrated preferences for what in Western music we call consonant musical intervals and simple songs made up of those intervals. But we can hear from about halfway through gestation in the womb. The auditory system is functional from about month 5 forward. So these babies had been exposed, never mind the 4 months post-birth, which is a lot, but also a little bit prenatally. So I was interested in the innateness argument. I said, well, we have premature infants. They're like minus 1 month old. They're like minus 2 months old. So why don't we see if we can look at whether they're sensitive to music in terms of heart rate or oxygenation or respiration after a heel stick. Now, for those of you who have done an experiment, it's like a great paradigm because they're like a petri dish or a rat. They're hooked up to a heart monitor. They're in an incubator. It's a very controlled environment. The heel stick is an event, so time 0 is very clear, right? And what we did was, at time 0, for 10 minutes, nothing, which is the control. So we talk about randomized controlled experiments. When a baby would come to the level 2 nursery in the neonatology ward at Mass General, we flipped a coin. And, heads, they were controls, tails, they were going to get the treatment. So the controls were no music. And the treatment group I chose lullabies because they have both speech or song and musical instruments. The tempo was moderate, the arrangement was simple, it was very consonant, it was, made sense ethologically in Boston to play lullabies. And what we found was that if we played music for 10 minutes after the heel stick, their heart rate was significantly lower than if we didn't play music. Another reason it's a good paradigm is because when they get stabbed, the heart rate goes through the roof, right? So you have a nice measure that you can tap into to see if there's an effect on heart rate. So their heart rate goes through the roof immediately after skin puncture, but if you average the heart rate over the next 10 minutes, it's significantly lower if you play the music than if you don't. This is some of the work that was funded by the Grammy Foundation. It's in Music and Medicine this year. The next step that we want to do is try to explore what types of music might work, what might not, because that would get even a little closer to the innateness question. For example, are there innate preferences for harmonic combinations of frequencies that make up consonant musical intervals as opposed to inharmonic combinations? Or is it just an auditory teddy bear effect? Like you could play anything, but as long as they're distracted by sound, or with the mother's speech, you can actually get their sounds recorded in the womb. So, women have signed up and had microphones put through the cervix into the uterus, and minutes of recordings have been made. So you can even play, womb-filtered sounds to the baby and see if that works better than music. So it's a track that we really want to develop. The really important part of it is that, there are like hundreds of thousands of babies around the world getting stabbed, and nobody's doing anything about it. So if we can develop a protocol that's easy for nurses to use, or devices that are easy for nurses to use that can deliver the sound within the incubator, we could really be helping lots and lots of babies in terms of their pain and stress that they experience in the first days of their lives. Now, a Japanese group who was working on drug development for hypertension, but they were interested in music. So there are rats that are congenitally breeded to be spontaneously hypertensive. And so different chemicals are tried to bring down that rat's blood pressure in this sort of step 1 or 2, phase 1 or 2 in drug development. So what they decided to do was at night for 2 hours to play music and to see if the rats' blood pressure came down, and then to do all their really cutting-edge molecular biochemical manipulations of the rat's cardiovascular system to get at if music is affecting heart rate and blood pressure, how is it doing it? What receptor systems? What chemicals are important? So here's the rat in his little box here. Here's the loudspeaker. You can probably see something in the rat. It's actually a cannula going into the ventricles of the brain so you can inject chemicals. And so they would play some music to the rat. Okay. That's the wrong sound file. That's the rat singing. Okay, how many of you recognize that song? Yeah, so that's Smashing Pumpkins, right? They actually played Mozart Divertimento 16, one of these typical kind of Mozart kinds of experiments. Maybe not for any good reason, but what they showed was that if they played Mozart for 2 hours, this is the systolic blood pressure measurement on the y-axis. This is time on the x-axis. And this is the baseline, right, without the music. So you can see the systolic blood pressure came down in the rats with the Mozart. But the really interesting part of the study from a neuroscience perspective is they then injected chemicals into the ventricles of the brain that blocks specific receptors in the brain. And they showed chemical specificity to the effect. So if they injected drugs that blocked receptors in the basal ganglia for dopamine, the dopamine D1 receptor subtype, and they blocked that receptor, it didn't matter. But if they blocked the D2 receptor, the music effect was abolished. And then they went on to do immunofluorescent chemistry to look at the concentration of different chemicals and receptors. And what they found was that, the color here has totally been aliased, but that a region of the basal ganglia here seemed to be an area with a high concentration of dopamine D2 receptors that were responsible for the effect of music on blood pressure. So it was a very specific biochemical effect that they showed music. It wasn't just some general effect. It was very specific to the D2 type of receptor in the basal ganglia. So to close, I want to say that it's at least been over the past 2 decades, neuroscience research in humans as well as animals has advanced basic knowledge about brain mechanisms mediating music, cognition, and emotion. The long, rich history of clinical experience in music therapy, including anecdote and introspection, that all provides a very rich source for generating testable hypotheses about how to use scientific knowledge to help sick children and adults, a major theme of our conferences. And that the confluence of scientific advances and clinical insights holds great promise for a new era in music-related research. Some people are trying to stay away from music therapy as a term, including some leading music therapists like Joanne Lowy and Suzanne Hanser, because it carries sort of all of this semantic baggage with it and methodological baggage with it. But there is a lot of potential, and it is starting to incorporate rigorous data collection and analysis methods using randomized controlled clinical trial designs. So again, thank you all for your attention. Thank you to the organizers for inviting me. And thanks to my colleagues for their contributions to the research and the support of the research through the Institute. Thank you.